give an applause again. Our next speaker there's a change in the program. One of the speakers are late, so
Aggie should come first. Because logically, I am to do
logically, but anybody will come for his presentation, that would be upside down. So free check, and you will take. That's what Beat asked from me. So our next speaker it's always you almost always in the last speaker. So
yes.
It's a moment of that. There's a very important ethical questions which we rarely talk about.
So what do I do to get?
It will be projected in a second. We have one spare. Thank you very much.
Okay. My name is Donald Bruce, and I run my own company in Edinburgh looking at ethics and technology called Edinethics. And in Edinburgh we have a big river just to the north of us called the Firth of Forth, and there are two bridges, the famous railway bridge, the Forth Bridge, and the road bridge that was built some years ago, which is now corroding, and they're building a new one, the so-called Queensferry Crossing. And I want to talk about building bridges in the sense of one of the big issues in the whole area that we're dealing with is the transition from animal to human and how we bridge that particular gap, which raises a whole range of ethical questions in the area. That's the company I am running, and we are basically the consultants, and if anyone has any projects that they have wished for an ethicist, I am here for business. I am currently working in the NanoAthero project, which is a 5-year EC project with 16 partners in 7 countries, trying to develop nanoparticle systems to transport and target contrast agents and therapeutic agents to key sites in vulnerable plaque and thrombus, blood clots, to treat atherosclerosis. Oops, have I gone? Okay. And it's very challenging, the idea of creating nanoparticles that will be able to predict that you have unstable plaque in your arteries and you are likely to get a heart attack and a stroke. And this is proving pretty challenging, as I will mention shortly. It could have a very significant effect if we can detect what is in fact the major cause of death and disablement in Europe. From an ethical point of view, it's a goal very much worth one's best efforts, addressing human suffering, reducing the burden on carers, reducing strains on the healthcare systems, and also reducing economic loss. 3 or 4 really, really strong goods. So what's an ethicist doing working in this project? Well, I'm working with the project as it develops, giving advice as necessary. And one of the issues is, in a way, the idea of the concept that you start off with, nanosystems, trying them out in the laboratory, trying to see which ones are working, which ones have the right characteristics, what materials, what ways of attaching ligands, and so forth. And you finally come to preclinical trials, which are supposed to take you across the waters into the point where you can try them on humans. And that's the problem. We've got some bridges built in other areas. This area has proved very difficult. How good is our bridge? Nanosystems are powerful, but that power can do harm. And so how to not put your materials where you don't want them, how to have effects that they are to have and not the ones you don't want, and so on. How do you validate your bridge sufficiently? And ethics, as I said, is accompanying this particular project. Well, preclinical trials, you're testing for efficacy, toxicity, side effects, distribution in the body, long-term fate of nanoparticles, whether or not they pass barriers you didn't intend as the ones you did intend. So how valid? We have 2 examples in NanoAthero. One is the ApoE mouse, which is attempting to model atherosclerosis. As we have gone through the project developing nanosystems, we've realized that this model is not very good. There are aspects about the permeability of plaques that we have discovered that the mouse simply doesn't do what human patients do. And it raises the question, how close do our models need to be? And the only other species we've got available to us is a rabbit, and that's not that close. We also have a different set of tests for a particular problem of complement activation, the body's total rejection of the system, which can have absolutely devastating effect. And Janos Szybeny and his team at Budapest, who won the Gnome last night, he has developed a CARPA test for complement activation and uses pigs. These are much more sensitive than humans. And you see sometimes a really dramatic effect upon the pig of injecting a nanosystem. I was actually present at one of the tests, and the pig just went into complete overload in this particular case. Only 1% of humans would have that reaction, but nonetheless, it's that 1% you're trying to avoid having it. And of course, we have had tragedies both in the UK and in France in recent years where what looked okay at the preclinical phase had devastating effects in phase 1. What happens if one of those happens in a nanoparticle trial? It's not happened, but how would we respond? Would the media say, nanotechnology, that's the cause? Or would the media be able to say, actually, this is a generic problem in all tests? You may guess that there is at least a fear that nano would be blamed. Should we move to large animals? In the development of genome editing technologies, the Roslin Institute, just outside Edinburgh, famous for the place where Dolly the sheep was cloned, they're developing the idea of, can you knock out genes in big animals like sheep or pigs that will be much closer than in some case mice would be, for example. So, when have we done enough to bridge to humans? And there are competing voices. The regulators say, well, we need to identify risks with reasonable certainty, but the patients say it's taking too long, I need a cure. The lawyers are saying, well, it's not so much safety, it's avoiding liability for the institution, the company, whatever. The shareholders, it's avoiding reputational damage, the share price goes down. For the media, it's either sensation or scandal. It doesn't matter which. And academics, publish or perish. You need to get that paper in a high score journal. Ethics. Well, here you're trying to balance animals and patients, and also research ethics, the tendency to overclaim your results, and the temptation to falsify, just to cut a corner somewhere, and it is happening too much. What about alternatives to animals? Historically, human cells haven't been all that good, often not enough cells, not enough the right types. I was in a project called SNATs where we're looking at human embryonic stem cells as a potential unlimited source of all kinds of cell types, but of course highly controversial. Can you replace one ethical problem of animals by another one, embryonic stem cells? iPS cells may be the answer, if they can work as well, but no one knows that yet. But cell cultures in general don't represent whole organs or the whole organism. So how do you address that problem? Well, one possibility is organoids, growing from stem cells, perhaps 3D printing, artificial organs, so-called, maybe bits of organs. Genome-edited kidney cells being grown into organoids are kind of being tried out as a test bed. So maybe this is going to be part of the way the future should be. But how do you test the model? How do you validate it? What would it actually model? What won't it model? So if you have to verify it, you still have to use an animal test system to check it out, presumably. So maybe it doesn't solve all the problems. At best, all of these are only going to give you partial solutions. Another idea is using animals themselves. Genome editing has got a number of possibilities arising recently for diabetes treatments. Growing a human pancreas in a pig, having deleted the pig pancreas instructions in the DNA. Also, an immune response edited pig islets that you could transplant into humans. So a number of possibilities there, but there are significant risk issues, crossing species. What about the ethical side of this? Xenotransplantation was much discussed in the 1990s, and we were heavily involved in that ourselves. Would people want a pig organ transplant or a pancreas grown in the pig? Well, it's not clear we'd necessarily want that. People are, Joel Haddow in Edinburgh University is looking at people's attitudes towards having something foreign in their body. It's research ongoing, but it'd be interesting to see what the results are. Some people dismiss the kind of yuck factor, the sort of revulsion, but I think don't miss a significant intuition. Because revulsion and aesthetics are not just about things we're not familiar with. They could be actually reaching down to something more profound in our ethical viewpoints. Perhaps there are cross-species ethical issues here. And also, should we be removing substantial parts of a pig's immune system? What is the effect on my sense of identity if I have an animal organ inside me? These are not insignificant questions. And are patients' perceptions different from the genome editors? The scientist sees, here's a real solution, but do the patients actually want the solution? There needs to be some checking here, I think. Finally, genome editing of human embryos. You may know that in the UK, an application was made to the Human Fertilisation and Embryology Authority, which accepted it, to do basic research in human embryos by deleting genes. The trouble is that research valid if you can't verify it in the end? Because you can't actually produce genome editing with a baby without the baby becoming an experimental subject. It's very different from animals. So in this area, I wonder whether there is a problem with the whole area of gene editing human embryos. Personally, I'm not saying it could never be done, but how would you validate it becomes a major, major question. So, I'm going to skip the last bit in the interest of time, but if you can't verify preclinically without making experimental babies, have we gone, as it were, a bridge too far? So.
Thank you very much. Questions, please.
Yeah.
I'm very happy to hear that you're still in Scotland.
Yeah? Well, Scotland is still in Europe at the moment, we hope.
Please stay.
Yeah, thank you. Thank you very much.
Just I couldn't help but thinking of this when you mentioned the yuck factor.
Yeah.
You said there's a certain reluctance in accepting big organs or implants, whatever. Does this reluctance or hesitation go down even to like a physiological response in the patient? Has this ever been looked at? Against the big organ that transplanted into, do I activate my autoimmune system and have that rejected?
I'm not sure. I mean, the only area I can think of would be using pig valves, which are kind of dead, just kind of there as dead tissue. I'm not sure you've actually ever had a live organ transplanted into humans. There have been experiments in primates of various sorts. The trouble is you get this. One of the problems, you get this enormous risk of the complement activation that is a kind of the total body rejection and how to overcome that. So you tend to see that problem anyway, and whether if that happened, it would mask any other effect, I have no idea. But I'm not sure that anyone's looked at the kind of physiological effect. But since we are, as both the previous speakers have indicated, we are complete whole beings, then to say it would not have some sort of physiological effect, I think it would be very unlikely that there would be no physiological effect if there was a sort of psychological one. I would imagine surely there would be, but no, I don't think anyone's ever tested that yet.
So you didn't mention computer modeling?
No, indeed not. Yeah, I mean, in silico as an alternative, certainly. But obviously, that is only ever a model. And you can take it, I mean, that will, in a sense, hopefully, focus your area into which area you should do or not do. But it won't actually ever, I don't think, be an alternative as such.
Yeah, let me make one brief comment to that. The problem with the computer modeling and electronic mouse and all those things is that there is artificial intelligence, but there is no artificial creativity. So that's not a dynamic system. Anything we put in our knowledge, but this doesn't guarantee that it's actually going to describe. I'm not saying we shouldn't do it. We should try everything that is possible. But the same thing, I'll just give you another example. you know the pieces of a chess, you know the rules, but you cannot foretell how a game between two persons, between a situation is going to go. So that's still not enough, it's still in infancy, but it's certainly one of the directions that at least the waste can be reduced.
Yeah, I mean, indeed, I mean, as I said, they're all partial solutions, and I think you need to have a combination that works the best in your system, recognizing you will always have this problem that you haven't actually modeled the patient. When we're discussing the question of it's very difficult to induce atherosclerosis in animals. And the 2 or 3 examples that are known, I mean, you think, well, why not a pig, for example? If you feed a very bad Western diet to a pig, the pig just loves it. Has no effect at all on the atherosclerosis. So you have a real problem there, or perhaps not a problem. But the problem then is also within humans. Okay, so suppose you've got your model, but human A and human B have really different effects. So how far is your model that is kind of the idealized situation actually going to be applicable to your range of patients?
Well, thank you very much.
Okay.
It's very interesting. I heard it this morning that the